Hello everybody, my name is CHIJIOKE and I work with the National Root Crops Research Institute, Umudike in Nigeria. I'll be walking through the presentation on yam quality evaluations at the National Root Crops Research Institute, Umudike, Nigeria. I'll be making this presentation on behalf of WP2RTB Foods team. For this presentation, we'll look at the different product profiles that we work on, the analysis we carry out on yam within the institute, some researches we've conducted and results, and the next level. At NRCRI, we concentrate on two product profiles from yam, the boiled and pounded yam. However, we also analyze samples from freshly harvested yams as well as dried yam chips. We measure some quality attributes on these product profiles. Some of them are chemical properties. We look at the pasting properties, the functional properties. We look at the cooking time for boiled yam, pounding time for the pounded yam. We also carry out sensory analysis on both the boiled yam and pounded yam using trained panelists. We do some instrumental texture analysis using either the texture analyzer or the penetrometer. And we also work on the quality traits of these materials, especially the fresh materials, using the handheld mirrors. The protocols we use are those that have been developed and harmonized under RTB Foods project. The first research we are looking at is titled Effects of Location and Genotype on Quality Attributes of Steam-Cooked Stored Alata and Rotundata species harvested from 2 locations in southeast Nigeria. For this study, we used 11 yam genotypes for both Alata and Rotundata species. These were harvested from Uyo and Umuahia in southeast Nigeria. These materials were stored for 3 months before we conducted analysis on them. For this study, we selected 3 tubers per genotype and measured their length. We cut each tuber into the head, the middle, and the tail, and washed, and then peeled and washed. For the different portions we removed from the proximal and distal end, we cut off 1/10 and 3/10 of the bottom and head sections of these parts and discarded them. While for the central part, we removed 2/10 from both ends of the section and discarded. Penetrometer test was carried out on each and on the remaining portion of the parts, and these served as representatives of each part. And then we also cut it longitudinally for steam cooking and subsequent analysis. We measured the cooking time. We used 4 pieces of yam per genotype for the steam cooking, and this to get the time for cooking the yam properly, we tested at 5-minute interval using a spec. The method we adopted was that was developed by FSA UAC Benin, and we used the steam cooking method. The results we obtained from this study shows that the cooking time for Alata species is directly is affected significantly by the clone in question, and we found out that the softness which was recorded using the penetrometer is affected by the clones. The pounding time is highly affected by the location, the clone, the section, and an interaction of both the clone and the section. For dry matter content, we found out that the dry matter content of Alata genotypes are related to the location and is also influenced by the clone and an interaction of the clone and the section. while the amylopectin content of the different section is related to the location and the clone, as well as an interaction of the clone and the section. Amylose for the different sections we found out is influenced by the clone and the interaction of the clone and the section. From the study, we found out that there may be a relationship between the pounding time and the amylopectin content of the yam species. For this study, we found out that the middle part, which had higher amylopectin content, we had to take more. It required more pounding time than the other sections of the clones. For the rotundata clones, we found out that for dry matter, the head region had higher dry matter content, and we found out that the dry matter content is also strongly influenced by the clone, the location, and the section being evaluated, while the amylose content is influenced by the clone and the section. Same for amylopectin. For the degree of softness, we found out that the clone and the section has strong effect on the softness of rotundata yam, while pounding time is influenced by the section. From this study, we found out that there may be a relationship between pounding time and dry matter. The study showed that the head region, which had higher dry matter content, required longer pounding time. The next study we are looking at is the quality attributes of sections of freshly harvested yam genotypes. This time we are using another method of cooking, which is boiling in water. For the study, we collected 13 yam clones from 2 locations in southeast Nigeria, Anambra State and Ebonyi State. We used a standard quantity of water to boil all the yam samples, 820 ml of water. And after boiling, we collected the remaining water. This was to ascertain the quantity of water absorbed by the different yam sections. And then we took processing parameters of the yam samples during cooking and pounding. We also evaluated the dry matter content of the different sections of the yam using standard methods developed under RTBfoods. The results obtained showed that the head region had higher dry matter content compared to the other sections of the yam. We also found out that the time required to boil the head section was higher. However, we found out that the quantity of water absorbed by the tail portion was more compared to the other sections, and that the time required to cook the head section was also more compared to the other sections of the yam. However, we didn't see more significant effect between the pounding time of the head section and the tail section. For the softness, we saw that the tail section was softer compared to the other sections of the yam. And then we found out that the color change for the tail section was also higher compared to the other sections of the yam. We also noted that the type of clone had significant effect on the rate of changing of color. We looked at the change colors of different yam sections for different yam genotypes 2 minutes after peeling. I mentioned earlier that we use the handheld mirrors to also carry out some quality trait evaluation at NRCRI. And one of the studies we carried out was to evaluate the possibility of using the handheld mirrors to measure dry matter content of yam tubers. For this study, we used 16 clones of deer letter collected from African yam germplasm at Umudike. The study was done using 3 different sizes of yam per genotype. The yams were washed, air-dried, and cut into 3 sections, head, middle, and tail. And we collected spectra on the different sections of the cut yam. The scan sections were chopped into smaller sizes and oven dried at 45 degrees for wet lab analysis. The spectra collected was transformed using the ViewSpec Pro software, and the wavelength we used to collect the spectra was between 350 to 2500 nanometer. The spectra was subjected to pre-treatment to correct any the results obtained show that the handheld NIRS can be used for predicting dry matter content of yam tubers. The R-squared value we got was 0.98, which shows that the NIRS can be used as a high throughput method for evaluating dry matter content of yam. The next level, we want to ascertain if there is a relationship between the sweetness of yam clones and species and cooking time of the yam. This comes up as a result of the survey that was conducted by WP1. We found out that there is this speculation that the cooking time of yam is related to the sweetness. So we want to conduct a proof of concept study to validate this information. We also will want to continue developing and calibrating equations to measure other quality traits using the handheld NIRS. We also want to look at the relationship between the biophysical properties of fresh yam tubers and sensory attributes of boiled and pounded yam. We appreciate NRCRI management team for creating a conducive environment that enabled us to carry out our work. We also want to thank African Yam Project for providing the materials that we used to carry out the research studies. We want to thank RTB Foods Project for involving NRCRI in this new area of research. And we appreciate the Bill and Melinda Gates Foundation for funding the work. I want to use this opportunity to thank my amiable team for all the sleepless nights and efforts they made to ensure that this presentation was possible. God bless us all. Thank you for listening.